Welcome to biology class. After pollination part, now we are in the pollen pistil interaction. Pollen pistil interaction, you know that the pollen grain it enters inside the pistil, and after that it will fertilize. So pollination, you know that is the transfer only. Pollination is transfer of pollen grain and over stigmatic surface. It but it does not guarantee the transfer the right type of the pollen. You know that when stigma is exposed in nature, then any kind of the pollen which is carried by the insect or air, uh, it it may fall over it and may germinate over it, but it may be some different other plants the pollen grain, so that there are some the compatible pollen and incompatible this terms here we have to use. uh, it means that here you can see that pollen pollen may be wrong type, either it is from the other species, it can be same species, but here the self incompatibility is there. It means that the same plant, but it will not fertilize. Okay. So that in these case that pollen will either it will not germinate. If it will germinate, then pollen tube will form, but pollen tube will not enter inside the ovule. It will not fertilize. Okay. Now come here, basic of it. I think in lower classes you have learned that. Here the pollen, when it will germinate, you know the pollen tube will form. You can see that pollen tube. Pollen tube is formed by the tube nucleus. You can see that the tube nucleus, and the male nucleus, when it enters in the tube nucleus, it divide into two. You can see that the male nucleus 1 and male nucleus 2, and after that when it will reach to the egg, one of the male nucleus will fertilize the egg. So egg, it turns into embryo, and another male nucleus fertilize with the polar nucleus, and this polar nucleus and the male nucleus that turns into 3N, which is endosperm. Endosperm is the food of embryo. So this is the basic. I think everybody should know. Now come here the different steps here, the pollen pistil interaction, and the pollination. At first the pollination is transferred the pollen. The pollen, if it is compatible, and the pollen addition will be there, then all the pollen grain less hydrated, so hydration will take place. Then pollen germinate, the pollen tube entry into the stigma, pollen tube growth through the style, pollen tube entry into the ovule and the embryo sac, and the event double fertilization. The double fertilization, just I discussed that the fertilization in both endosperm and embryo will form that one. So we can see this fact of the pollen pistil interaction. Okay, pollen addition. It means the pollen it sticks over the stigmatic surface. You can see that here it is pollen grain, and the chemical, the protein, lipid, the pollen coat, and the protein on the stigmatic surface, this contribute the addition. It require the protein interaction. In this way that they can survive for the long time. It will not even germinate. In some cereals, as a rice, wheat, the pollen grain, it lose their viabilities within 30 minutes, but in some other cases it stays for a month. So in this way that will the addition takes place. No, the pollen will hydrate. Hydrate because it has 15 to 35% is there. So, so here it takes the water, takes water on the surface of the stigma. So and hydrate and ready for germination. Next, pollen tube germinates. Look at here this tube. It will germinate that the tube nucleus. The pollen grain, the enzyme like alkaline phosphate, ribonucleosesterase, amylase, all these are, it helps in germination. The pollen tube contains the enzyme cutinase, which dissolve the style, the area. We can dissolve that and get enter inside it. GABA and cysteine rich protein have also shown the function in the pollen tube guidance. You know that when pollen tube will reach to the synergids area, I mean inside the ovule, will get the entry. If still it is not compatible, then it is stopped there, and pollen can sperm cannot enter inside and reach to the ovum. So next part we will see that. Look at here that pollen tube is reached there, the male gametes. This first one you can see. Here this the synergids. This is the egg. This is the egg, and this is the pollen nucleus, which is 2N. See that here the filiform apparatus, here the present in the micro pilar part of the synergids. It guides the entry of the pollen tube. The entry of the pollen tube, it guides. So when they enter, you can see that the sequential stages. Here it entered inside. Here the male gametes, it is released from the tube. So this is the filiform which helps this to move the male gamete. Now male gamete is moving. In the third one, the male gamete is moving one towards. Look at here. Look at this two, the egg and the one male gamete number one. And look at here another one. This is polar nucleus and male gamete number two. So they're going towards that, and the union will take place, and the fertilization also takes place. I think you know that this polar nucleus and the male gamete, it will form the 3N cell, which is in plant it is quite common. In animal it is not there. And here in egg and the another male nucleus, it will form the embryo, and this plant will grow from that. So pollen and the pollen pistil interaction up to here. In the next part we have to see that the double fertilization, that development after that. Post fertilization event, structure and the event, and the embryo and endosperm, how it will form, and the latter part of it. Till then, thank you.